Now we are going to talk about chromosome structure. Chromosomes are different in prokaryotes and in eukaryotes. In prokaryotes, the DNA is shorter than in the eukaryotes. It is one circle and it is not associated with proteins. So the prokaryotic cell does not have chromosomes, it has one piece of circular DNA. In eukaryotic cells, the DNA molecules are longer, they form lines and not circles, and they are associated with proteins called histones, and this is how they form the chromosomes. In eukaryotes, since we have mitochondria and chloroplasts, which we don't have in prokaryotes, the mitochondria and chloroplasts contain their own DNA. This DNA in the mitochondria and in the chloroplasts is like the DNA of prokaryotes. It is short, circular, and not associated with proteins. So, chromosome structure. Chromosomes are only visible as distinct structures when the cell is dividing. At the start of cell division, they become thick and visible. They appear as two threads joined at one point called the centromere. Every thread is a chromatid. It is a replicated DNA. The DNA in every cell, if we lay it out next to each other, is around 2 meters in length. However, how does it fit inside the nucleus of a cell? It is highly coiled and folded. So you can see this is the DNA double helix. It is then coiled and combined with histones to further compact and further coil on each other, and then the coils form loops, and then the loops themselves coil and pack. So we have different levels of compaction for the DNA to fit inside the nucleus. The number of chromosomes varies from one species to another. In humans, we have 46 chromosomes, potatoes have 48, dogs have 78, and so on. Keep in mind that in most species, we have an even number of chromosomes in adult cells.